Hello everyone. In this lecture, we are going to discuss about the innate immunity, which is otherwise known as natural immunity or the native immunity. That means whenever the body encounters a foreign pathogen or a foreign antigen, innate immunity is going to provide the first line of defense. Now let us discuss what are the components or what are the cell types that are involved in innate immunity. The first one is epithelial lining of skin and gastrointestinal and respiratory tract. These epithelial lining, their predominant function is to provide the mechanical barrier. Okay, so these are going to provide mechanical barrier property against the foreign pathogens. But apart from this, these are also going to produce certain proteins which will have antimicrobial properties. These are known as defensins. Okay, so these epithelial linings, they are going to produce some proteins which are known as defensins, and these defensins will have antimicrobial properties. Also, these epithelial linings are going to have certain lymphocytes which will play a role in this first line of defense. Okay, though we have discussed lymphocytes play a major role in the adaptive immune system, but some lymphocytes which are there in the epithelial linings, they are going to have innate immune function. The second group of cells include neutrophils and monocytes. So basically, these neutrophils and macrophages, they are going to have phagocytic functions. They will have this phagocytic function. The macrophages, which are present in the tissues, okay, these are nothing but the tissue phagocytic cells, macrophages. These macrophages can be derived from the monocytes that are present in the peripheral blood, but some of the tissue resident macrophages are also derived from the yolk sac of developing embryo. Okay, so these things we have discussed when we discussed about the normal hematopoiesis and the process of phagocytosis, and more on these things we are going to discuss under the section of inflammation. The third type of cell is dendritic cells. Dendritic cells are professional antigen presenting cells which will have surface projections in the form of dendrites. That is why the name dendritic cells. They have a dominant role in the adaptive immune system, as we have discussed previously. They are going to present peptide antigens to the T lymphocytes. Okay. However, these dendritic cells also have certain properties pertaining to innate immune system. They are going to produce certain cytokines. Once they encounter the antigen, they're going to produce certain cytokines, and these cytokines help in the process of inflammation against the particular pathogen. Okay. The next type of cell is known as the innate lymphoid cell. So what do you mean by innate lymphoid cell? So this means tissue resident. These are tissue resident lymphocyte-like cells. Okay. Lymphoid means lymphocyte-like. These are lymphocyte-like cells. Why these are known as lymphocyte-like cells? Because some of their functions are similar to the lymphocytes. However, they are not lymphocytes because they do not have the lymphocyte receptors. They do not have lymphocyte receptors. That means the T cell receptors or the B cell receptors. However, some of their functions are similar to lymphocyte. That is why these are known as the lymphocyte-like cells or the lymphoid cells. One of the important example under this heading you can remember is NK cells or the natural killer cells. Okay, so natural killer cells are sometimes described under this heading of innate lymphoid cells. More on this we are going to discuss in subsequent slides. There are certain other cell types like the mast cells, endothelial cells. They also have a role in this innate immunity. More on mast cells we are going to discuss when we discuss about the hypersensitivity reactions. Plasma proteins like the complement proteins. They are going to play a dominant role in this innate immune system, and these things, the details of complement proteins and the entire complement system, the complement pathways, all these things we have discussed in a separate video in detail. Okay, so I'm not going to discuss about this in this lecture because we have already discussed this. Also, there are some proteins like lung surfactants that are there in the type 2 pneumocytes of the lungs. They also have innate immune system functions. Okay, so these are the components, these are the cell types that are involved in the innate immunity. Now we are going to discuss about the receptors that are they are in the innate immune system. That means the cells of innate immune system, they are going to have certain receptors to identify the foreign antigens. Okay, so if you see, this is a cell of innate immune system. This can be, this can be dendritic cell, it can be the macrophages, it can be the neutrophils. So basically, these cells of innate immune system, they are going to have certain receptors. These receptors are known as pattern recognition receptors. Okay, PRRs, or pattern recognition receptors. Okay. What do you mean by pattern recognition receptors? These are pattern recognition receptors. These are not specific for a particular antigen. These are not specific for a particular type of antigen. This is in contrast to what we have discussed for the lymphocyte receptors under the adaptive immune system. We have discussed that the lymphocyte, one particular lymphocyte is going to have a particular type of lymphocyte receptor, and that particular receptor is going to identify only a particular type of antigen. However, pattern recognition receptors, these are not specific for a particular type of antigen, but these are specific for particular type of patterns. These are specific for patterns. These patterns are shared among the related microbes. Okay, that means multiple microbes can have a particular type of pattern which can be identified by this innate immune system. Okay, and these pathogen associated patterns are known as PAMs. PAMs are pathogen associated molecular patterns, and these receptors can also identify some of the molecular patterns that are present in the cells, that are present in the products of a damaged cell. Okay, so if you see, this is a damaged cell, it is going to produce certain antigens. These antigens are going to have a particular type of pattern. Those are known as DAMs, damage associated molecular patterns. Okay, so these pattern recognition receptors, they are going to identify either the PAMs or the DAMs. Okay, now we'll discuss in detail about this pattern recognition receptors. The first type of receptor is the toll-like receptor. Why it is known as toll-like receptor? Because the founder member or the first member of this group is toll receptor, which is described in Drosophila fly, and in Drosophila fly, it is going to have function in the development of the fly. Okay, these toll-like receptors, these are either present on the cell membrane, or this can also be present on the endosomal membrane. Okay, so this is the endosome. Okay, so these toll-like receptors, some of the toll-like receptors, like the TLR2, 4, and 5, they are present on the cell membrane. And because they are present on the cell membrane, they are going to identify, or they are going to recognize the bacterial cellular products. However, some of the toll-like receptors, those are present on the endosomes, they are going to recognize the genomic components. Okay, they are going to recognize the genomic component of the pathogens. Okay, so if you see the TLR2, it is going to identify peptidoglycans. TLR4 is going to identify lipopolysaccharide, and TLR5 is going to identify the flagellins. There are around 10 types of toll-like receptors. These are some of the examples, but these are important examples. The toll-like receptor 2 is going to identify peptidoglycans, 4 is going to identify lipopolysaccharide, 5 is going to identify flagellins. But the toll-like receptors that are present on the endosome, they are going to identify the genomic components. Okay, so basically TLR3 is going to identify double-stranded RNA, TLR7 is going to identify single-stranded RNA, and TLR9 is going to identify CpG DNA. Okay, so once the toll-like receptor is engaged, once they are activated, they are going to activate two important transcription factors. Okay, and most of these toll-like receptors, they are going to act through a particular pathway that is known as mid-88 pathway. Okay, so most of the toll-like receptor, they will have downstream mid-88 pathway, and through this pathway, they are going to activate the genes of two important transcription factors. So what are those two transcription factors? Those are NF kappa B, nuclear factor kappa B, and interferon regulatory factor. Okay. So they are going to activate two important transcription factors, NF kappa B and interferon regulatory factor. NF kappa B is going to produce cytokines and adhesion molecules that help in the process of inflammation against the particular pathogen. And interferon regulatory factor is going to produce interferon alpha. And this interferon alpha, this has antiviral properties. This has antiviral properties. Okay. So this is about the toll-like receptor. The important point that we have to remember is toll-like receptors can be present on the cell membrane. They can also be present in the endosomal membrane. Those that are present in the cell membrane, they are going to identify the microbial products that are present in the cell wall. And the endosomal toll-like receptor are going to identify the nuclear material of the pathogen. Okay, once they are activated, they're going to activate two important transcription factors through mediate pathway. I mean, not always, but most of the times they're going to act through this pathway, and they produce NF kappa B and interferon regulatory factor. NF kappa B is going to produce cytokines and adhesion molecules, which help in the process of inflammation. Interferon regulatory factor is going to produce interferon alpha, which has antiviral properties. So this is about the toll-like receptor. Now we are going to discuss about the second type of receptor, that is known as nod-like receptor. Nod-like receptor, that means the founder member of this group is NOD2. Okay, it is NOD2. In contrast to the toll-like receptor, nod-like receptors, these are intracellular cytoplasmic receptors. Okay, so if you see here, this is the nod-like receptor. This nod-like receptor, it can identify bacterial products. It can identify bacterial products, both intracellular and extracellular bacterial products. Okay, it can identify reactive oxygen species. It can be activated by potassium efflux. It can also be activated by various crystals like uric acid crystals. Okay, and also it can be activated by extracellular ATP molecules. Okay, so all of them, they can actually activate the nod-like receptor. Once the nod-like receptor is activated, it is going to activate another protein complex, multi-protein complex, that is known as inflammasome. Okay, so once nod-like receptor is activated, it is going to activate a multi-protein complex that is known as inflammasome. This multi-protein complex, usually they have three important components. One is the NLRP3, that is leucine-rich protein 3. They will also have adapter protein, but most importantly, they will have this caspase 1 in the inactive form. Okay, so this inflammasome, which is a multi-protein complex, it is composed of NLRP3, adapter protein, and caspase 1 in the inactive form. Once the nod-like receptor is activated, it is going to activate this inflammasome. Once this inflammasome is activated, it is going to produce active caspase 1. Okay, so it is going to release active form of the caspase 1. Then what is the function of active form of caspase 1? It is going to convert the precursor form of interleukin 1 beta to the active form of interleukin 1 beta. Okay, so active caspase 1 is going to convert pro interleukin 1 beta to interleukin 1 beta, and this interleukin 1 beta will be released into the it will be released to the systemic circulation, and it is going to produce the inflammatory response against the particular antigen. Okay, so this is how nod-like receptor is going to act. Now we'll discuss some clinical significance of this type of receptors. The first one is what is going to happen if there is a gain of function mutation. If there is a gain of function mutation of the nod-like receptor. So from this discussion, you can understand that if there is a gain of function mutation, it is going to produce unregulated interleukin 1 beta. And if there is increased production of interleukin 1 beta, it is going to have inflammatory or systemic inflammatory response without any antigenic stimulation. Okay, so that is why this condition is known as auto-inflammatory syndrome. Okay, so gain of function mutation of nod-like receptor is going to produce auto-inflammatory syndrome, and of course, the treatment has to be antagonist of interleukin 1 beta. Okay, so interleukin 1 beta antagonist is the treatment for auto-inflammatory syndrome. The second important clinical significance is some of the diseases are associated with inflammatory response produced by this nod-like receptors. For example, if if they produce response, inflammatory response against the urate crystals, they are going to produce gout. Okay, it is going to be associated with gout. Similarly, if they produce inflammatory response against the cholesterol crystals, this increases the risk of atherosclerosis. Okay. Similarly, against lipid, if they're going to produce inflammation, that is associated with obesity-related type 2 diabetes mellitus. Okay. So various disease conditions are associated with inflammatory response produced by activation of these NOD-like receptors. Okay. So this is about the second type of receptor that is known as NOD-like receptor. Now we are going to the other types of receptors. The other type of receptor, the first receptor is the C-type lectin receptor. Okay, so this is the C-type lectin receptor. It is usually present in the dendritic cells and the macrophages. Okay, so C-type lectin receptor, it is also a cell membrane receptor, as you can see from this diagram. It is going to identify fungal glycans. C-type lectin receptor is going to identify fungal glycans and it is going to produce inflammatory response against the fungus. Okay, and these are present in the dendritic cells and macrophages. Now coming to the second type of receptor, that is known as the Rig-like receptor. Okay, the next type of receptor is the Rig-like receptor. This is the Rig-like receptor. This Rig-like receptor, it is again intracellular cytosolic receptor, similar to what we discussed for NOD-like receptor. This Rig-like receptor, it is going to identify the viral DNA. These are going to identify the viral DNA molecule. And once they are activated, they are going to activate a particular pathway that is known as STING pathway. And through this STING pathway, they are going to produce various interferon molecules. They are going to produce various interferon molecules, and these interferon molecules will have antiviral properties. Okay. So Rig-like receptors, they will identify the viral DNA molecules. Once they are activated, they are going to activate the STING pathway. Through this STING pathway, there will be production of interferons that will have antiviral properties. And if there is a gain of function mutation, which can cause increased activity of this STING pathway, it is going to produce a particular type of disorders. Those are known as interferonopathies. Okay, that means there will be increased production of these interferons. Okay, and that is named as interferonopathies. Now coming to the next type of receptor, these are G protein coupled receptors. As we all know, G protein coupled receptors are 7 transmembrane receptors. These G protein coupled receptors, these are described for neutrophils and macrophages. And these receptors, they are going to identify the N-formyl methionine residues. Okay, it is going to identify N-formyl methionine residues. Okay, this N-formyl methionine residues are going to present in all the bacterial proteins, and some of the mammalian proteins that are produced inside the mitochondria. Okay, some of the mitochondrial proteins will have this N-formyl methionine residues, but all the bacterial proteins, they are going to have this N-formyl methionine residues. This can be identified by the G protein coupled receptors, which are present predominantly in the neutrophils and monocytes, neutrophils and macrophages, and activation of this receptor is going to help in the process of chemotaxis. Okay. Now coming to the last type of receptor that we are going to discuss is the mannose receptor. This mannose receptor are going to identify mannose residues that are present on the various bacterial proteins. Okay, so these are going to identify mannose residues present on bacterial proteins. Okay, human proteins doesn't have mannose residues, but bacterial proteins do have mannose residues, and they can be identified by these mannose receptors. Okay, so these are all the receptors that are involved in the process of. These are all actually they are component of pattern recognition receptors, and they play a significant role for identification of pathogens in the innate immune system. Okay. Coming to the next section, now we are going to discuss a little bit about the natural killer cells. Natural killer cells, these are actually lymphocytes, and in the peripheral blood, around 5 to 10% of the lymphocytes are NK cells. Okay, 5 to 10% of the lymphocytes are NK cells or the natural killer cells. As we have discussed previously, around 60 to 70% are T lymphocytes, around 20 to 30% are B lymphocytes, and around 5 to 10% are NK cells. Okay, and they are going to kill the abnormal or the stressed cells. Okay, so they are going to identify and kill the abnormal and stressed cells. And what is the mechanism? How they can actually help clearing all these cells? Those things we will discuss now. So these natural killer cells, they are going to have various types of receptors on their cell surface. So what are the important receptors? The first one is CD16. They will also have CD56. Actually, CD16 and CD56, these are markers of natural killer cells. Along with this, they are going to have certain activating receptors. They're going to have activating receptors, and they will also have inhibitory receptors. If the natural killer cell, if the activating receptor of the NK cell is activated, it is going to activate the NK cells. If the inhibitory receptors are activated, that is going to inhibit the natural killer cells. But what is the function of this CD16? So basically, CD16, the CD16, it is going to act as receptor for immunoglobulin G. Okay, so if you see, this is the immunoglobulin G. This CD16, it is actually a receptor for Fc component of immunoglobulin G. And this immunoglobulin G, as we have discussed previously, this has a opsonizing property. That means they can bind to certain antigens and help in opsonization or phagocytosis of those antigens. Once these NK cells, they can identify this immunoglobulin G, which has opsonized a particular antigen, it can actually come and destroy that particular antigen. Okay, so once NK cells engage their CD16 with the immunoglobulin G molecules, they can actually destroy that particular antigen, which is opsonized by the immunoglobulin G. Okay, and this process is known as antibody-dependent cellular cytotoxicity. Okay, because this cellular cytotoxicity is dependent on the antibody, that is immunoglobulin G. Okay, so this process is known as antibody-dependent cellular cytotoxicity. So NK cells are going to have this property, and this is because they have the CD16 receptor, which is a Fc receptor for immunoglobulin G. Okay, now coming to the second scenario. So if you see, this is an NK cell. So NK cell will have activating receptor and inhibitory receptors. Once NK cells encounter a normal cell of the body, as we all know, all the nucleated cells of our body, they're going to have the MHC class I molecules. And sometimes these MHC class I molecules, they can present the self antigens to the NK cells or sometimes to the T cells also, that we have discussed in the adaptive immune system. Okay, so basically the inhibitory receptor of the NK cells, they can identify the MHC class I molecules. Okay, they can identify the MHC class I molecules. So once the normal cell presents the self antigen with the MHC class I molecule to the inhibitory receptor of NK cell, the inhibitory receptor is going to be activated. Once the inhibitory receptor is activated, it is going to inhibit the NK cell. It is going to inhibit the NK cell. That means NK cell will not damage this particular cell. It will not damage the particular cell. That is the self cell. Okay. So in this process, the NK cell can identify the self cell, and it is not going to produce any kind of immune response against that particular cell. However, if the cell is abnormal, for example, if it is a virus infected cell, or it's a neoplastic cell, it's a virus infected cell, or it's a neoplastic cell, these cells, they are going to downregulate. Some of these cells are going to downregulate the MHC molecule, MHC class I molecule. If they will downregulate the MHC class I molecule, the NK cell cannot engage its inhibitory receptor. On the other hand, these abnormal cells, they are going to produce certain, present certain antigens through different types of receptor, which can actually activate the activating receptor of the NK cell. Okay, so I hope it is clear. The abnormal cells, they are going to downregulate the MHC class 1 molecule. If they downregulate the MHC class 1 molecule, the inhibitor receptor of NK cell will not be activated. On the other hand, they will present some of the abnormal antigens which can actually engage the activating receptor of NK cell and activate the activating receptor. Once this receptor is activated, it is going to activate the NK cells. It is going to activate the NK cells. Once the NK cell is activated, it is going to release some proteins like porphyrins and granzymes, and it is going to damage or destroy this cell, destroy this particular cell. Okay. In this process, there are various cytokines that are involved, like interleukin 2 and interleukin 15. Interleukin 2 and 15. Yes, interleukin 2 and 15. They help in proliferation. Interleukin 2 and 15. They help in proliferation of these NK cells. And interleukin 12, it helps in activation and increased killing by these NK cells. Okay, so these are the cytokines that are involved in proliferation and activation of the NK cells. Okay, so 2 and 15, they help in proliferation. Okay, apart from this, sometimes the NK cells are going to produce certain cytokines like interferon gamma, which will activate. As we all know, interferon gamma, the predominant function of interferon gamma is to activate and recruit the macrophages, and these macrophages can come and they can phagocytose the abnormal cell. Okay, so these are the functions, these are the properties and functions of natural killer cells. Okay, so natural killer cell is constantly surveying the cells that are present in our body. If it is a normal cell, it will engage its inhibitor receptor and it is not going to cause any harm to the normal cell. However, if the cell is abnormal, it is going to activate, it is going to engage its activating receptor and it is going to activate itself and produce damaging molecules. It can activate the macrophage so that it can clear the abnormal cell. Okay, so now we have discussed about the cells of the innate immune system, about the receptors of innate immune system. Now we'll see in a nutshell what are the reactions of innate immunity. The predominant function is inflammation, because as we have discussed, these innate immune cells, they are going to produce various cytokines and also they have properties of phagocytosis. So in this process, they are going to produce inflammation. Inflammation has, of course, there are predominantly two functions. One is the elimination of microbes, but along with that, they also help in the process of tissue repair. All these things we'll discuss under the section of inflammation again in detail. Then the next thing is they will also have these antiviral properties, especially the interferons or interferon alpha predominantly. Interferon alpha, it is going to have antiviral properties, as we have discussed that these interferons can be produced, especially if the Reg-like receptor is activated through the STING pathway, and also when the toll-like receptor is activated through activating the interferon regulatory factors, it is going to produce the interferon alpha. Okay, and these interferons are going to have the antiviral properties. But one of the most important function of innate immunity is linked to adaptive immunity. Okay, these things we will discuss in detail when we study about the adaptive immunity. Okay. Now coming to the last section, that is how innate immunity is different from the adaptive immunity. I mean, there are so many differences. Like as we have discussed, the cells are different, the receptors are different, how they function, those things are different. So all those things I'm not going to write them again, because once we study the basics of adaptive and innate immune system, we have already understood all those things. The three important property that I want to discuss here is, the first one is specificity, second one is timing, and third one is memory. Innate immune system is not specific. It is not specific. This is because they do not have specific receptor for a particular antigen. As we have discussed, innate immune system is going to have pattern recognition receptor. Those receptors are not specific. That is why innate immunity is not specific. It's non-specific. Second one is timing. Of course, it has to be rapid because this is the first line of defense. In contrast to the adaptive immune system, which is not rapid, but innate immune system is rapid. And what about memory? Of course, innate immune system is not going to have memory. Okay, so memory is a property of adaptive immune system, but that is not the property of innate immune system. Okay, so that is all about the innate immune system.